So thanks for the invitation and also for the introduction. So as I put in the title, like it's a nice title for the DIP because the Institute for Emergence Phenomena. So I thought, okay, I should talk about my ongoing work. So this work is based in my research fellowship at IES. So that started a few months ago, like last year, let's say. So it's not done, but I always prefer to be more excited about things I'm doing than things I did. But if I will mention a few things I did before, but there's more in progress. So if anyone can give feedback, I really appreciate because it's under development. Yeah, so a small presentation about the team I belong now. So I am at the Department of Anatomy and Neuroscience at the Amsterdam MC. So I'm in the multi-scale neuroscience department. So it's led by Linda Dow here, and so I'm here. So it's a very interdisciplinary team from physicists like myself and applied mathematicians to psychologists. So it's all range of professionals because we do research in clinical neuroscience. So you have to like include patients, interview patients, and also build networks from patients, things like that. So you need a very interdisciplinary team. I will tell a bit what I do there in one slide, but I will focus in the IAS fellowship. So when I proposed to make this fellowship, I wanted to ask like fundamental questions like everyone in IAS in a way, so that I wanted to better understand complex systems. So like most people here, I would say. I would say that the brain would be the most important complex system for multiple reasons and the most complex. So it would be a kind of empirical setting that's really nice to explore. And then like we are developing lots of methodologies, but the first empirical data set that we are going to apply is the brain, more specifically brain imaging in functional MRI. Yeah, so that's kind of to narrow a bit more. Yeah, so the whole fellowship will be based in brain image data. We have lots of brain image data depending on temporal resolution that you want or spatial resolution. In our lab, we work in EEG, the electroencephalogram, MEG, magnetic encephalogram, and MRI. So then these are the type of images that we use, but for today, we stay in MRI. But in the lab, we actually build what is so-called multi-layer network. So this is the work I was involved, like the mathematics part, let's say like that, to create from a single individual a multi-layer network where you have different images from like EEG, MEG, and fMRI, and create a unique network of networks. So that's the one single slide about the work there. But if you want to talk about this, you can talk also later. But that's like in multi-layer networks. But for the IES fellowship, because the mathematics and the methods are a bit new to neuroscience, we focus in one modality first. So, and the focus from the IES fellowship will be in functional MRI. So often we build this, we have these both signals in an MRI machine, and you compute some similarity measures that could be a Pearson correlation, mutual information, coherence, multiple similarity measures. And with those similarity measures, you would build a network. So this is the starting point, let's say, for physicists, applied mathematicians to understand the brain. Like it's often a team. Oh, yes, please.
So first of all, like, why do you think like a network is the right kind of tool to understand brain? And then what does it even mean to like understand brain?
Yeah, it's very good question. So there's no accurate answer, and also non-methodology are good to understand the brain, in my opinion. That everyone tries everything it's possible, but if you go to a microscopy person, he will get samples of tissue, but this is really accurate in terms of tracing out the neurons, but you cannot trace the full brain because there are like 8 billion 100 billions of neurons, so you cannot trace. And you go to imaging, each modality traces different scales in the brain. So if you go to functional MRI, it traces really nice spatial scales, so you can have a great spatial resolution, but really bad with time resolution. So if you want that modality, you know what's good, what's bad. So for fMRI, spatial scale is good, time scale is exactly bad, and the activity is really fast time scale, so you cannot really grasp. But if you believe in a functional resting state functional MRI, some sort of ergodicity, so that you could have a longer time series when you acquire, like minimum like 10 minutes, but some people acquire like a brain scan in an MRI like for half an hour. You could think about some ergodicity, and now you can think that there is functional connectivity, like the two areas are synchronized at a high correlation, then you can assume that. But in fact, you totally right, and I don't think there's any good tool to encode information about the brain, but it's super relevant. I think we could be the next pandemic, like in 50 years, everyone will be 100 and with Alzheimer dementia, so we need to start the brain. So that's like, thanks for the question, but I feel like, yeah, that's what the one I can help, but there's no unique. So I don't know if that answers your.
So like the kind of short-term goal is to like take some signals from the brain and understand some correlation. Is that like
That would be the past short-term goal. Here, I try to find emergence, and I try to put this signal in a better formalism, at least for a theoretical physicist. But in short, long story short, yeah, you process the signal from a brain scan. So there's no invasive, because you cannot, for ethical reasons, we cannot just open humans' brains. But yeah, so we process the signal from the fMRI. And what they proposed in this fellowship was do a multivariate signal processing that would create hypergraphs and simplicial complex from the image. So that's more like, if I go to more details, it would be like that. But if I go, is this the best or not? I agree that there are multiple ways, and some are good depending on the time scale or the spatial scale that you are interested in, the phenomena that you want to model, but none are perfect. That would be my view. I don't know if. Great. Okay, thanks.
Another question: are you focusing on brain fMRI scans of healthy individuals, or also on people with some sort of brain disease, or Alzheimer's, or?
That's a nice part of working in the neuroscience department. So the product now is on healthy individuals, so as to calibrate the metrics, kind of. And but in the team, we study a lot the glioma individuals, people also with multiple sclerosis, epilepsy. So these are the three diseases in the team that I work, they approach more often. But of course, there is also Alzheimer or other diseases that are relevant. But for these, like as I'm anticipating, so I try to create a high order metrics that are meaningful or emergent for the brain. Then we first calibrate in healthy individuals. What I show now is 400 healthy individuals, young adults, so not even with variability in the age. But the long-term goal would be to apply in disease, like in disease cohorts as well. And with that, I have worked in that, but in other topics.
Why do you focus on resting state?
So that's the kind of the resting state would be the first stage. Like there is also two kind of branches in network neuroscience for fMRI, the task-based MRI and the rest state MRI. But for calibrating some new stuff, let's say new mathematics for neuroscience, I think would be more like wise to start with resting state, so to see how is the brain at rest. So I think it is based also in the results. But if you put more tasks, then the hypotheses are more neurobiological. So you need to come with more insights from neuroscience. And here we just have an insight that if you have a long time acquisition, so in your brain scan, you stay very long enough to assume that some sort of ergodicity, and what you measure are correlations that are functional correlations. So if you think about resting state, it would be to create such a network. But as I say, the time scales of the fMRI is too long when you compare to neural activity.
Yeah.
So if you want to do dynamics, maybe it's better MEG or EEG, or you have to have a different calibrate your time scale so that you observe a change in the dynamics of the brain. But yeah, that's also a good question. But yeah, that there was like, let's say, the basic choices for set up a new metrics in neuroscience that will be our start. Is it,
Yeah, I'm just wondering, we'll see in the slide, but I'm just wondering how you're going to link this, because resting state is notoriously difficult to also understand, right? This is,
yeah,
drift can happen, you can think about something for 10 minutes and then you pick it up in the scan where somebody else can do something totally different. Whereas task-based would be more accurate, and EEG or MEG would then give you more temporal dynamics that you can link to states. And with fMRI, there's also a bunch of stuff with like the hemo response can lead to downscale activation, even though the actual activation of the brain areas go upriver, if you will. So fMRI in general is very difficult signal to work with.
yeah, I agree. Hopefully, you should tell me your opinion after the talk again. Like if you feel like, okay, there are also other problems. Like if you go to EMGs, okay, because I'm talking about MRI, it's nice to raise the issues of fMRI, but EMG, there's a huge problem in source reconstruction. So you don't know the spatial scales. So for physicists like audience, so you do a image reconstruction. So you get the signal and you assume that there is a dipole in the brain that generates your image, the magnetic signal. And this problem has multiple solutions. So you actually not even know whether you observe where is the signal in the brain. So the time scale of imaging is really nice. So you have like 1,200 points per second. It's really in the time scale of neural activity, but the spatial scale, it's difficult to trust. So you would not know from where this signal comes from if you want to do really more theoretical stuff. So as I say, it relates to the first question. Sorry, what's your name? That each modality has good and bad sides, and then I recognize all the bad sides of fMRI. I try to make in a way that is okay. But if I would go to another modality because of the issues on fMRI, I would also find other issues. For EMG, you have a spatial, like really lack of spatial resolution, and multiple methods would yield different outcomes. So you'd have different spatial information about your activity. So it's also tough. Then I don't know if that's
No, I know, I'm familiar, so I recognize this too.
yeah. But yeah, so I want your opinion after the talk also. Okay, so then anticipated the common approach is based on creating some similarity metrics. So you compute, like, let's say, Pearson correlation, coherence, phase lag index, so different indexes that would quantify functional interactions between areas of the brain. And that would build this brain network. That's a functional brain network. And then I make this illustration like where I put the strongest links first, so you can have different connectivity in different areas of the brain. But for most people, this is the standard network approach. But if you think about the brain, so I don't know too much about the neuroanatomy of the brain, but if you think that there are about 150,000 kilometers of axons in the brain, making metrics that are POIs maybe are not the ideal. So that's the hypothesis. So the working hypothesis, this slide summarizes the working hypothesis of the fellowship, is that in most of 3 points, interactions are approximated by the sum of POIs interactions. So you make these similarity metrics only between pairs of the areas of the brain, where actually the brain is so complex that it could be that 2, 3, 4, 5 areas of the brain are talking to each other at the same time. And then we can assume that the brain has a network not the same as the brain has a hypergraph or a simplicial complex. So if you get multi-body interactions in the brain, you may unveil different patterns in the brain, or emergent patterns in the brain. So that would be the main hypothesis, and then we have to create some sort of methodological approach. So how to build a signal processing pipeline where this is taken into account. So the common pipeline, if you go to this slide, you throw in, let's say, in your code to create your brain network, mirror image, 2 time series, and then you check whether there is strong relationship, similarity between these 2 areas, and you get a link. So you have to create a similar pipeline for multi-body interactions. So that's kind of the methods part. This is for any complex system that is kind of based in signals, let's say like that. But then the nice part of the brain and the human connectome project, you can replicate this in multiple people. So then each person is like a stochastic realization of such thing. So then we think like, oh, the brain is a really good empirical system to test this. Yeah, so these plots were made like in a work that we did recently. But why I'm doing that? So now that relates also to my previous life. Let's say like that, I started as a theoretical physicist, and my PhD was relating statistical mechanics and topology and phase transitions. And there are lots of results relating differential topology and classical phase transitions. That was my inspiration for start with this. But actually, what is, I can add this, I think some of you already saw this animation, but you can think a network is just a projection of a high order structure or space. So if you now talk about the brain, so if you assume that each of these circles are brains, you can still differentiate if you think as a network. They are planar structures like PYs, like a planar, geometric is like a planar structure. And okay, maybe 5 different colors, a bit different thickness, and etc. But if you think this is a high order structure, like a hypergraph or simplicial complex, there are infinitely many different ways to project a hyperdimensional surface into a circle. So then when you play this video, you only realize that they're actually totally different. So that's the reason, like the theoretical reason, okay, if I create high order representation of brain image data, either patterns that we know about the brain may be emergent patterns, or you have, like if you apply the methodology to different groups, you may find we spot a difference between the groups easily. Like it's a methodological paradigm, but if it works, can be actually beneficial. So,
but let's say you only constrain like these higher dimensional objects to fit according to the biological constraints. Is that the gist of it? So you have these high dimensional representations of your data.
Yeah.
And that can be many shapes, they can all be viewed as a circle. But you then want to know which high dimensional object you should look at.
Yeah, exactly. So in terms of if you make an analog with networks, so if you start only links in a network, many different networks could be seen as the same network.
Yeah.
And if you say, oh no, I want to start my data has a hypergraph or has a simplicial complex, then 2 different networks could be 2 similar networks within the pairwise framework, may are different networks in a high order framework. So, and it's tough to illustrate that, but when you do in geometry, it's easier. Oh, everything that's projected in the plane has millions of ways to be in space.
Right.
So then it's like, oh, then it's a good analogy if you don't to skip the maths, but that's the reason. Like if you make a high order representation, you may find a lot of difference. And but my personal inspiration was also the relation that these may have with physics. So if you go through string theory or supersymmetry or these other theories in physics, that many of the things are explained by topological invariance. So that Euler characteristics, Petty curves, I don't know, these T-Singer theorems. There are lots of topological invariants that are, in the end of the day, explaining a lot of physics. And if you represent data, like network data, as a simplicial complex or hypergraphs, you actually can compute those invariants. So it's more of an analogy kind of agenda. And there are many people working hypergraphs and simply complex in real data, but many of them are based in mathematics. So what's the best way to represent and stuff? But what you should look, let's say, this is a really difficult task to create a hypergraph from a brain data. Once you have that, what you should do with this? So we also have to give some direction, and then the one way is to follow some things that were done in physics. It's like that was like first work I did in this direction was 2 years ago. That was, I think I presented you in this presentation, Casper, on the phase transition. So if you assume that topological invariant Euler characteristics could predict a phase transition in a network, that's actually true. So, and if you know the literature in physics, there are many papers, not always not unique, but there are many papers where this invariant also predict phase transitions in statistical mechanics. So you can just say, oh, actually you want to build a high order representation of the data of a network. You have an agenda which is test whether principles of theoretical physics could be observable or have some analogs in data. So in brain networks, we did it 2 years ago. We made some sort of claim that this could happen in different complex systems. That was like, oh, that's a bit, let's say, speculative. But since then, there was applications with this idea, like in financial systems, in other biological systems, protein networks, genetic expression networks. So it's for this example, was a good case. I don't know if for other analogies that would be a good case, but that's for me was the reason. Oh, we should maybe look for a really good high order representation of network.
Sorry, can I ask you something?
Yes.
So you just mentioned this topology, being some sort of quantity that you could use for understanding phase transitions just in normal physical systems. I mean, besides the brain networks, I'm just wondering how do you get topology in normal, I don't know, canonical ensemble for systems that we know in physics? Where is topology coming from?
So you can do use Moss theory. You can get your Hamiltonian, let's say in a classical Hamiltonian. So you have your energy level surface, like then for every iso surface, you can study the topology of the potential surface. And then I think the mathematical branch is like in differential topology. Like if you get a continuous system, then you have to go through differential topology, and then you can trace how topological invariants change when you change your measuring your ensemble. This is more done in like a microcanonical ensemble. So you fix your energy, and for every energy level, you start the topology of your system. And then there's.
But you mean but which invariant? I mean, you're talking about something like a Chern number associated with a band, like the work you just showed by the physicists, the quantum physicists.
No, in this specific subfield, the main invariant is the Euler characteristics.
Yeah. So can I ask a question related to this, and also answering Jay's question? I mean, you're actually looking for disconnected subspaces, not so subvolumes.
In high dimensions.
In high dimensions.
Yeah.
And so after, if your temperature or your energy is too low, then you just you cannot reach all parts of space, and you lose aridicity actually.
Yeah.
And this is what changes the topology.
Yeah.
Is that clear? That's true, no?
Yeah. So yeah, maybe okay. I can zoom in a bit more. So it's exactly what Peter just said. Like you can have a equipotential surface, and like in this illustration, like for the torus, and then when you slice the torus, nothing happens, and there will be a critical point where there is a major change in the topology, and then that will be related to a phase transition. So then you can trace a different for every energy level, you can trace or compute the topological invariants, and they have often markers of transition. So it's theoretically nice because it's independent of a measure. So you don't need to create a statistical ensemble. Only the topology of your manifold, like of your Hamiltonian, the manifold that derived by your Hamiltonian could explain phase transition. This is not one-to-one, but there are a few theorems. If you want literature on that, I can share after
I
this.
think it's a great measure, but it might be very difficult to get. In a complex system, this is not easy.
In a complex system, there is a way. It's combinatoric. Combinatoric is complex, but to build a simple complex, you just need a polytope, like a high-order polyhedra. So for Euler characteristics, it's a combinatorial problem. So every click in a network is a phase. So if you count all the clicks of a network, you have a topological invariant, which is the Euler characteristics. So it's combinatorically it's expensive. So you need to fix a maximum click size, let's say, but it's feasible.
Yeah, but okay. So in terms of high dimensional spaces, you need to be able to get the entire network, the entire.
Yeah, for brain networks, it's feasible because if you have a network like 100 nodes, you have 2 to 100 clicks. It's computationally feasible, but it's in the limit of non-feasible because the enumeration of those clicks is NP. So then it's not feasible. But for the brain, it works really nice. I only put one slide, but basically these gray curves are like 500 different individuals in the human connectome, and the blue is like the average, and then they follow like some sort of phase transition patterns, which are generalizations of percolation. So geometrically, if you increase your parameter, that's like the correlation level that you can attach different links, there will be a critical threshold where there is a percolation. So any point A and B in the brain would be connected by these links, and that would be this first transition. If you do that for simplices, like for triangles, tetrahedra, there will be always a different threshold where there will be percolation, but then now a percolation of simplices, not a percolation of links. So I made a video on this. So it's not then you can see like in the top is just the nodes, in the top right is like the links, like the way you see, and in the top left are the three clicks and the 4 clicks. Like then each one of those they percolate, and if you compute the Euler characteristics, you trace each zero of the Euler characteristics basically is a marker for each percolation. So it's really, but it was out of the theory. Like it was not like I didn't came that with that from the brain, but from the maths. And when I didn't put it in this slide, but if you test it for disease cohort, if you think that, oh, these are like critical temperatures, let's say like that, then water, if something boils at 100 degrees, it's very likely to be water if it's at the same condition. So if you have like 180 degrees, could be oil or something like that. So if you test different cohorts, they have different critical points. So it's a theoretical way to make a distinction between groups. So in this work, I didn't put the plots here, but we show that glioma patients, they have different phase transitions. So if you just put next to each other plots, they have different phase transitions.
I mean, I don't understand something. In a physical system, you have, of course, energy or temperature, which you change. But what do you change here? The number of links per the degree, the node degree?
Yeah, you can change the edge density because the function network is a bit arbitrary. So we don't know what's the right amount of links that the function network must have. And then in applied topology, a common procedure is to test all possible densities. Yeah. So that's when you go to this video, basically I'm changing the density. So
but this is actually a random network. Actually using random, you place links randomly.
No, I place the strong links first. So the links with high correlation, the 2 areas that are strongly correlated are first in this process called filtration. So we start with the empty brain like here, and then you put the high correlated links first, the second highest, and so on. So this parameter tells how strong should be the correlation. So if it's 0, no correlation, 0, 0, 1, 99% correlation, we can put the links, and then here 85, 70, 50, and so on. So this procedure is called like filtration. So you test all possible correlation levels that you should include to create your brain network. It's a big problem in network neuroscience because each medical group chooses a different density. So then they don't know, oh, should I put 5% of the strongest links, the 10% of strongest links? So in a way, this kind of solves this issue because we actually test all correlations, and then you build like some nested chain of networks. So you put one network like that has like in the illustration tells for itself. Is it clear?
Yeah, that's very clear.
Ah,
Thanks.
thank
So you have a critical density where the percolating threshold.
Yeah, and this you can trace with one topological invariant. So the Euler characteristics trace all the transitions for in every dimension.
So you get a kind of a hierarchical picture of the whole
Yeah,
thing, like
yeah.
it's nested.
Yeah.
Right.
So, sorry, can I speak something?
Yes, please.
Yeah. So we actually we could understand the parameter at the threshold on the interaction matrix. Am I right?
So say again. So I didn't understand.
Yeah. So we could understand the parameter here, 0.0, 0.01, something like a thresholding on the interaction matrix.
Exactly. So in terms of
Okay.
what is happening behind the scenes, like you have a empty matrix, and then you start to put the threshold in the connectivity matrix from the top links first until the weakest links.
I see. Thanks.
Thanks for helping.
So now let's say you have this and you want to make some like experimental prediction. What is the prediction?
So you can compare between groups. That's what's happening in different domains. So you can, if you have a cohort with a disease or cohort is healthy, you could check the difference between groups. And that in this scheme, after we published this paper, someone made similar analysis for attention deficit disorder. So for different diseases, this could be a pattern. So each disease would have a different critical point. This is a global analysis. In a local level, we can even know a bit more because topology connects with geometry. So I skipped this part because I wanted to go to the high order representation, but you have the Gauss-Bonnet theorem also works for networks. So if you want to project it locally, every time you compute the Euler characteristic for a network, you can compute curvature in each node of a network, and then you know locally what's going on. So for this work specifically in attention deficit disorder, they could trace which areas were different, just tracing local counterparts. So we also did that for glioma, but we didn't report details. But okay, there's a change in curvature in different areas. So we have current work that where we can compute curvature of frontal parietal networks from the default mode network, and they differ when you change for different cognitive levels. But this is ongoing, but I'm happy to share after. Don't know if that.
So in principle, you can, let's say, tell if someone is sick or not from that.
Yes, it's a fancy way to do that. Depending on the disease, you don't need, but for very subtle disease or for cognitive decline, for things that are really subtle in the brain, that's one direction. Not necessarily what I'm doing, but using topological data analysis for medical care is really a big trend in applications in medicine. So you can create these simplices or high order representation of medical data and make some prediction. They call it individualized medicine on that. But then it's still like, let's say, it's going through translation to a patient, but I don't know where that is being really applied. But in the hospital, we do similar things like to the patient, but we are in a medical school, not in a common hospital. Okay, so then
I have a short question. Did you apply it separately to pairwise and then 3-body and then 4-body, or did you do it when you do the 3-body, the 3-body click? Do you do it pairwise at the same time? I mean by that, do you add pairwise links at the same time that you add 3-body links, or do you just add on one hand only the pairwise and then on the other hand only the 3-body and then on the other hand only the 4-body?
That's a very nice question. Thanks, Claire. Like in the beginning, like this field that does this kind of approach called topological data analysis or topological neuroscience, And the first approaches are based on what Clelia just mentioned. So you add a 3-point interaction, at the same time you add a pairwise interactions. This is not necessarily true. That was like the beginning of the seminar. Say, okay, some of pairwise interactions are not necessarily a 3-point interaction, but still we have good results. This paper I showed you 3 years ago was under this framework, and the framework that I proposed for the IS fellowship is to process the signal directly. So the 3-point interactions observer directly from the 3 signals. So that then what I show now is independent. What I just showed is dependent. Thanks, Claire. Thanks, I didn't clarify that. Okay. So then the methodology is to quantify these interactions based on the signal, and then I would like to apply it in rest state fMRI. So that's long story short. And in later product, we want to relate these findings of this method to behavioral traits of individuals. So the nice thing of human connectome database is that you know many traits about individuals like age, sex, education, cognition, personality. So and the sample size is really large. Like we have database of about 1,000 individuals. You can really check whether those metrics, although they're abstract, they relate to anything in behavior. So that's a good end goal in terms of application, but there's no theory, motivation. So it was what I just told you. Right. Okay, and yeah. So far, we also want to quantify how the hubs are also relates to the work of Clelia. I think if some of you know her work, maybe it will be easy to navigate. Because there is a combinatorial problem, as I just mentioned. Like if you have a network with 100 areas, 100 links, like the 100 nodes, this is the case of these sort of brain networks. This is the length scale of the nodes. You have like, I don't know, 5,000 links. But if you have triplets, you have 125,000 triplets. So you have a really combinatorial issue. And then we have to solve this issue first. And that's why, okay, let's first try the hubs, because this decreases the complexity of the problem a lot. So that's what I present today. So IS mediate a lot of stuff, a lot of communication. Like we have events on how to make this methodology. So I want to thank Rick and a lot of other people that we made calls, discussions, workshops. I forgot many of them, but I put a list of the ones that fit here, like people that I had discussed at croissant coffee discussions. So lots of people that helped. I really thank a lot of them, and some of them involved already in some projects. Yeah, then after presenting the people that helped, I want to show what's this methodology. So in a way, so I give an example of 3-point interaction, but that works for n-point interaction. So you have a 3 time series, and what you want a similarity metric. So a metric that would say, quantify some connectivity between these 3 areas. And instead of having a connectivity matrix, you have some sort of connectivity tensor, and then you would create this high order network. So that's instead of having piecewise connectivity driving the network, you would create a network directly from the signal in the order that you would like to have your network. And that would be like the signal process pipeline that we want to test whether it works or it's good for brain network analysis. So that's kind of one illustration for one individual that made that pipeline. For people who are familiar with neuroanatomy, then you have primary areas that pop up first, like the back of the brain, more occipital areas, the vision, and the foot mode network. Let me put it again. And the central areas like motor areas and so on. So this makes a lot of sense. But I want to show then the behind the scenes. So basically, when you go to this methodology, if you think what's the best mathematical framework that would fit to capture such interactions, a natural choice is information theory, because it was since the 50s or 60s extended to multi-body interactions. So although it was never used extensively, there is an extension of information theory that is beyond PY. So if you have a multi information, when you have true signals, you can have interaction information or different metrics that would capture 3-point interactions. So basically, for every possible metric, you can get the connectivity. So then if you go to the intersection between the 3 signals, you will have interaction information, which is the extension of multi information. If you get all the pairs of connectivity, so it's total correlation. Or if you get the whole signal, it's a joint entropy, and so on. So there are many, there's a zoo of metrics that I maybe don't go into details now, but basically each one is a candidate for high order connectivity.
I have a question.
Yes.
Yeah, could you say something about what S information is? I'm not sure if I heard about that before.
Yeah, so I'm not the best person to say that. My training is more in topology, but S information is some sort of magnitude of your high order interactions, and the O information is more about synergy and the redundancy in the interaction. So the S is more like the strange, let's say an analog of strange, and the O is whether the interaction is synergetic or redundant. Like synergetic is more when it happens only in the high order level, and redundant is more like it could be seen as a sum of the parts. So a 3-point interaction that is a sum of 2-point interactions is more redundant, and a 3-point interaction that is exclusively 3-point is more synergetic.
Yeah, I see.
So
is the S information also found in these information diagrams like all the others, or is it?
No. As far as I know, no. Maybe you know more, but in these diagrams, it's easy to spot joint entropy, multi information, and total correlation. There are other fancy diagrams that maybe give that.
Yeah.
But
I think the O information can be found in these diagrams, but yeah, for S information, I don't know.
Yeah. Okay. So, but in the end of the day, thanks, Leon, for the comment. In the end of the day, you have spreadsheets per person. So in the end, we have this is a brain atlas, and there are 92 areas, and then I have 90 all possible triplets, that possible connectivity, and then for every possible metric, I quantify it. So it's normalized. Then you don't see the negative value like between 0 and 1, but basically I show you for all this, for one person, what's this distribution. So you have a lot of signal that is like process, like we optimized this process. So we did it for 100 individuals, and this is like behind the scenes what's going on. And then we have a big problem in the sense that I have 125,000 possible interactions. So here I think it has like 200 or so. So I cannot even represent. So that then we say, okay, maybe we have to find a way to see what are the most important interactions. That's why I mentioned, it's very close to the work of Clelia, because she starts like high order interaction more in spin systems, and she concluded, correct me if I'm wrong, Clelia, please, that when you go to multi-body interaction, there are not many relevant interactions. There are only a few of them are relevant, and in this framework, that's also going to happen. So then, what we have, like this is a same plot, but I'm not telling what's the metric. This is the illustration for starting for the strongest interaction information and putting new triplets at a time, how it looks like. So it looks like a primary function in the brain for people who are familiar with neuroscience, but it's just a lot. So we have to come to an approach that decreases the complexity. Like you create a lot of complexity processing the signal. Now we want to find to rank what are the most important. That's why I say high order hubs. So the most important triplets.
But so what are these nodes doing then?
So here it's an atlas, and I'm plotting each triplet at a time. So I have like 200 frames in this video, and then the strongest comes first. So it's a filtration based on the exchange, like the same.
But there's node sizes being changed, right?
Yeah, the nodes is proportional to the amount of triangles that you participate.
Okay.
Sorry, I didn't, like I just passed through it. But the node size tells you in how many, just to get a local information, in how many triplets are you involved.
Right.
So this is like more like the way to, that is commonly represented in neuroscience. So you know.
It's odd that the occipital cortex is the highest one.
Yes.
Would you expect that the parietal cortex would be one of the most connections?
If you increase the order of the interaction, it comes to the Pareto and to the default mode. But
Yeah.
if you think about primary functions, vision and motor.
Okay. Yeah.
So then but I will come to that if I have time. When it's time to stop, please tell me. I'm okay. Then we have to make some like I put again this video. When you go to high density, it's just uncountable. So we implemented a hub algorithm, so we can create a network. So this is not too much. We have 125,000 triplets. So we are very happy that the mathematics of high order is the same mathematics of pairwise interactions. So we can just study the connectivity matrix of this hypergraph, and we study the highest eigenvalue and highest eigenvector, and that tells us what are the most important triplets. That's like what we did. And then when I start doing that, I realized that any other tool like modularity, community, between centralities, diffusion, everything you could also do in this. So it's an endless agenda. So if this works, we have lots of work to do. But I start first with the hubness. So that's why I presented this today. But a full panorama of what is going on. So you get fMRI data, you get a multivariate information theory metric, do the signal processing, create a hypergraph. From that, it's just too much. Then from this hypergraph, you do Perron-Frobenius theorem or PCA to get the strongest triplets, and you get the hubs. So that's a pipeline. And okay, this is more for medical scholars. Like, oh, they afraid, oh, how you compute that is the same. So the same way the distance in 2D, 3D, and 4D compute similarly. If you have this high order network, all these metrics, if you know the algorithm part, the implementation is analog. So that's just to say I'm using the same metric. And we aim to do different high order metrics in the future, but now we just start with the hubs. So then I now go to the results and how we build this connectivity. So there's a lot of mathematics on building this connectivity, and this one I'm showing you is the one where the existence of hubs is guaranteed. So then I can send you reference if you want to dive into this more, but I put this really easy example where we have like 4 triangles. And obviously the central triangle is the most important, like you don't need to make a Perron-Frobenius theorem for that. But if you enumerate each triangle, let's say I put labels in it, then I have a connectivity matrix of triangles. Then the rule of connectivity that allows you for hubs is the rule where you can say that, okay, if a triangle shares an edge, they are connected. If 2 triangles share the same edge, they are connected. If you want to do it high dimensionally, so if you have a simplicity of dimension k, if they share a simplicity of dimension k minus 1, they are connected. So with this rule, you can always create a Genesis matrix. So then, sorry, the figure is not yet professional, but basically you have these 4 triangles, and then 1 and 3 are connected. So I go to line 3 and 1, and this 1. I go to line 2, like say 2 and 3, and it's connected, and so on. So we create this adjacency matrix. Of course, in the early stage, it's binarized. I could do weighted, I could do different variations. And I throw in the strongest links first, like the 1,000. I did up to 10,000 to check the robustness of the analysis, but I just throw in the 10,000 strongest triplets, and then I make this algorithm, like I create this connectivity matrix, and then run Perron-Frobenius algorithm, the largest eigenvalue and eigenvector, and then get the centrality, a normalized centrality. So then with these, it's like a to the force for this example, but then we prove that the B, D, and E has higher centrality. So that is nice for this example, but for the brain, I will have lots of triangles. So then it's better to use the algorithm in the brain than to visualize it. And that's what I show you now.
I have a question.
Yes.
Could you say something about what the eigenvector centrality means?
Yeah, so it's a name that is common in network science, but it's an application of Perron-Frobenius theorem to networks. So you get your adjacency matrix, and then it's known that the largest eigenvalue of the eigenvector of the adjacent matrix is like the principal component of your network, and then you define and normalize the eigenvector for this highest eigenvalue, and the coordinates of these normalized eigenvectors are the hubness of this node, of a node. So basically, you just start the highest eigenvector.
Yeah.
And
The principal component, what exactly does that mean?
So it's in principal component analysis. This is the first component. It's just when I talk to people that are not in networks, they don't know what centralities are. And when you go to principal component analysis, the first component is the component related to the maximum eigenvector. So it's more like try to explain different languages. But maybe here it's okay to try talk about hubness or centrality. But in networks is when you talk about centrality, there are different centralities that's more related to the degree, to the betweenness of a network. And the eigenvector centrality is the one related to the Frobenius theorem.
Okay, yeah, thanks.
Is it good? Thank you.
I have a
Sorry.
question.
Yeah, you go ahead. You go ahead, Oscar.
Okay, thanks. I was wondering, how do you, so each of the nodes here is a different area of the brain or different part of the brain where you have data? On this graph, each of the like A, B, C, D, they represent one of those areas?
Yeah, each HBCD is one area, but what I measure now is the 3-point interaction. So the nodes are now triangles.
And how do you, yes, okay, this I understand, but how do you put values on the triangle? Like, do you look at how often those ABC kind of are co-activated, or how do you put, let's say, yeah.
So you get 3 signals, and then you define a hyper edge by quantifying a multivariate information matrix. So you get.
I see. Okay.
So every hyper edge, every triangle is brought a value of the, let's say, the interaction information between 3 signals.
Yeah. Okay. Sorry.
So I understand your question, because in the past that would be, if you have 3 areas co-activated, it would be a triangle, or 3 links or 3 edges in a network, there would be a triangle. So what I'm proposing is that, because in PY's frameworks, the 2 signals give you the connectivity, then if you're in higher order framework, 3 signals would be a triangle connectivity, 4 signals, quadruplet or tetrahedral connectivity, and so on. So was it okay, Clara?
Yeah, so the measure is the mutual information, basically.
Yeah,
the 3-body mutual information. Okay.
Yeah, or not only mutual, but can be also, let me see, all the
other measure. Okay,
yes,
sorry, yeah.
These are all these 5. I test these 5. There are others. I just don't know enough information theory to test everything. But if anyone wants to help, I can test others. But yeah. But it's a problem. Like in network neuroscience, in our lab we use 14 metrics to define network. So like, oh my God, there's so many metrics. So each one could be relevant for different schemes. And for this work, we already test 5. So, but
how do you detect the cliques? Because they're, remember, from my maybe confusing to like a clustering coefficient, but with clustering coefficient, for example, there's different ways to compute them, and they all produce different results.
Yes.
But that's not for cliques, probably. I think I'm confused.
No, cliques is a combinatorial algorithm.
Exactly,
you just check if there is connectivity between the triplets or n-plets. And here we don't have cliques anymore. You have the strength of each triplet is defined by an information theoretical metric. So, but in the end of the day, instead of having, so we had to threshold, like we cannot put in the eigenvector centrality algorithm a matrix with 100,000 triangles, is still too much. So we put the 1,000 strongest ones, and we tested up to 10,000 to see whether it works. That's where I say it relates to the work of Clelia. Like in this example where you have like 1,000 strongest triplets, where I do the principal component of the eigenvector centrality, like 960 have 0 centrality. So they are not relevant. Only those small fraction, like about 50, are relevant. So most of the 3-point interactions are not relevant, which makes sense if you think in theoretical physics or the approach, like most of the nature is PYs, but there are a few high order interactions that are relevant. So it's nice. So let's then, this is the animation I showed before, and now I show one that's, say, in the left, you go all the triplets up to like 200, I don't know how many put, and in the right, only those ones. So that's one nice thing. So that's what's going on for this specific metric for interaction information.
And
So
it's asymmetric now.
it's asymmetric. You know why?
Well, this guy's moving to the, what is he doing? Moving to the left or
No, this is a motor area. So
Yeah.
in the center of the brain, the pre and post central areas are motor, and they are like asymmetric. So if you're right-handed,
Yeah.
it's stronger in the left and vice versa. And this cohort has like 100 people. It's the mean plot for those 100 people, and 90% are right-handed. So it should be left. But I anticipate the interpretation to tell your answer, but okay, I can tell. So this is what is we think is emergent. Like basically, we got these thousands of triplets, and what are the most relevant ones, and then you select them and plot into the brain atlas back without any prior assumption. And then when you plot it, okay, this is motor system. I didn't know that because I'm not neuroanatomist, but I'm 2 years in the department, so they brought me to this dissection class, anatomy class. Okay, this is.
But it's
this
resting state. Right?
Sorry?
It's resting state.
Rest state.
So how come the motor area is like that?
So if you're resting and I throw you boiling water, you can move. Your brain is aware about sensory and motor things. Like if you need something happens, even in rest, you can move. So your brain is not off. So your vision and your sensory motor areas must be on also in rest state. That's what I think. Maybe it's better to go to the next slide. Let me play again this. So it's one metric. It's this, but what I want to summarize it, each metric that we choose yields a emergent pattern that is compatible with something that we know in the brain. I cannot say it's a ground truth because we need to do more, but it's very promising in a way. So the interpretation was with help of this neuroanatomy. So he's a professor of neuroanatomy in my department. And then these are his classes slides. So these two areas that I show you is called, one is prefrontal gyrus and post-central gyrus, and they're together responsible for sensory motor tasks. So one part feels, the other one does stuff, moves. And it's known that this system is coupled. Like, okay, this is not the same, but if someone throws you hot water, you move your hand. So feeling the hot water is sensorial, moving is motor. This is automatic, you don't even need to go through your brain, but that's just an example to explain that sensory motor areas in the brain are coupled. So anything that you need to spread to the rest of the brain, then therefore must be three-body. You know, like, okay, the sensory motor are always talking to each other, they are near each other. There's this sulcus that separates them, but then they, in terms of neuroanatomy, basically they integrate everything with the rest of the brain to make some sort of sensory motor activity. So this was emergent. So that was a conclusion we had. And then when we did for each of those metrics, we got such pattern. Some were redundant, like two different metrics got similar pattern. I'm showing a second one because I think I'm now running out of time. But if you do the same, but now with total correlation, then you pop up vision. So this is a vision system. So in your occipital areas, you have a left and right part of the back of the brain are processing your visual stimuli, and you need to process it and spread over the rest of the brain. When you do the same process, like that, it's the 1,000 strongest total correlation triplets, and then you select only the strong ones, then this comes. So we are a bit scared in the beginning. Oh, is it possible or not? But it seems that you can always find it compatible with a specific system in the brain. And there is one theory. It's not theory if you tell to a theoretical physicist, but if you tell to a neuroscientist, it's a theory that's called like segregation integration theory about the brain. So the brain is integrated, so all the areas of the brain must be connected and share information to each other, and is segregated. That means that specific parts of the brain do specific tasks. So in one go, if you project it like that, you find these two patterns, because The big hubs like this, in this case, the vision areas are kind of processing vision stimuli, and they are spreading to the rest of the brain. And you see that in one go. You don't need to make it's like data-driven and emergent. So that was our first conclusion. So we now, so first I would like feedback from you guys, but we now run for a publication that without clinical insights, but we already start doing clinical analysis on that to check, well, what are the relationship between those hubs and what do you know about those people. But what I want to say, we did not consider any prior knowledge about the brain in our analysis. Each higher interdependency was compatible with some systems in the brain. I have videos from the others I can share if you're curious. Oh, I want to know joint entropy, I want to know whatever, I can open. But I think the two is nice, and these two are primary function, vision, motor, primary functions. That's really compatible. And then with this integration segregation patterns that I just told you that is a signature of the brain emerge spontaneously when you do it. And then we want to, in the future, use, so there was a summary. So we want to do different, like more either theoretical in one hand, if you want to do modeling on that. It's like Rick is interested in this direction as well, or clinical approach, like to check whether the traits that you have annotated on those individuals, they correlate with those hubs. I have one analysis that I have here, I can also show later. But before I stop, I can tell what comes next. So there was, I think this is on interest of many of you guys, because IS is sponsoring the use of a human connectome database. So it's really tough if you're not in a hospital or in a neuroimage group to have access to brain networks, because it requires a huge neuroimage preprocessing pipeline. So this database that you use, it's in about like 22 terabytes of data. And then you have to pre-process the signal, get each voxel, time series, and etc. So in the end of the day, then you have a network. And it also it's difficult to have collaborations for privacy rules. You cannot access data if you're not working in a hospital. So it's really tough. If you want to collaborate with neuroscientists, you have to work hard to circumvent these issues, and it's inherent from the field. It's not about personal anything, just happens like that. So then IS was very nice to us, and then sponsored two research assistants. They are they have master in neuroscience and AI, like the Mini, and Flores in neuroscience and philosophy. And they spend the last 4 months or 5 months downloading and organizing two big databases. So we're going to test these abstract calculations in 725 individuals between 36 and 100 years old to check if you have correlation between aging and those high order metrics. And there's another one that's going to be out, but this is coming first, whereas the ACP young adult. So we have 1,200 individuals between 22 and 35. And all these two databases, you have annotated many traits. So if you want to check whether this correlates to anything, is a good database. So we will book a day to show you guys how to use it or what you need. And we make available already networks. You don't need to use only for high order stuff, but if you want to do a normal network product with this data, we can share what is shareable. Like basically everything that's post-processed, that where you download and create something, you can share. So we cannot share the raw data, only if you get permission from the Human Connectome product. So we had to write a proposal and stuff. But if you say, oh, I want to use these individuals because I have a nice idea in agent-based models or in whatever network stuff you like, IS will have this. So it will be, I think, organize it with Rick, how we make it accessible, but soon that will be accessible. Yeah, that's another advertisement. And thank you so much. Sorry, maybe I run out of time, but thanks for the time.
Great. For this very interesting talk. I'm gonna see if there are some questions. So Marcus, you can just unmute and
Yes, thank you. Yes, so I'm working on higher order Laplacians and higher order diffusion in in network geometries, and I was wondering if these can be mapped to the brain in some way. So yes, my question, I guess, is do I understand correctly that the networks that you're constructing this way necessarily they are in a sense triangles that meet in edges? And if you would also combine this information or different metrics would you then still have networks that have this a structure like this, or will you then have also like triangles that will only meet in a node?
Yeah, well, some things like I don't know, someone is on but from.
We are speaking through that.
I'm muted. I'm muted. Is it better? No.
Your speaker.
Here's my
Turn off the speaker on your computer.
Is it better?
Yeah.
Yeah, there we go.
Yes.
Sorry, Marcus, for the issue.
No problem.
It's a really nice question, and yeah, we envision applications on high-order Laplacians, like the Hodge Laplacian and combinatorial Laplacian. And yeah, you understood correct. Like, when you build this high-order network, so there are two steps. One is defining high-order interactions. So it's a more empirical, like from how to jump from empirical data to a high-order structure. And the second one, once you have that, how to do the analysis. And there are multiple tricks on how to build a high-order network from high-order data. And a few of them allows to create high-order Laplacians. So both combinatorial Laplacians and Hodge Laplacians. So if you're interested, I'm happy to discuss, because there are lots of diffusion Laplacians. Also, there are like metrics that are based in diffusion, like between centrality is based in diffusion, was already extended to high-order framework. So you can have a between centrality within this high-order network. So yes, if you answer for questions, yes, and thanks for.
Yeah, so I guess it depends on how many different types of interactions and regions in the brain you include. You can get different types of networks that you study. So yeah, either ones that are like yeah. So I mean the different types where you either have for example, triangles that always meet in edges, or more general shapes that can also meet in only a point, let's say.
Yeah, you can create a rule for this one that I show was a rule for hypergraph, because I didn't define a full simplicial complex. This is also in the goal, but for the full simplicial complex, you would have to create new rules for this rule, the set rule for simplicial complex. Like any big simplicial complex must contain the smaller ones. So like if you have a simplest of dimension k, all the simplest of dimension k minus 1 should and smaller than that should be in the simplicial complex. So for that, you need to come with other rules that are meaningful for your empirical data. There is a work by Pierre Baudot that starts doing that. He helped also us. He came to give a talk in IES, but if you check the work of Pierre Baudot, I think it's a good start to check how you could build the simplicial complex for information. I think it's called information topology. If you Google information topology, you'll find his work. But yeah, it's a interesting avenue. But in a way, we started with the easiest, and we have a long-term agenda. Like you have to start with the simplest problem first, and if it's good, you go more and more. But one of the directions to build a more complex high-order representation that could be the simplicial complex, and then you have space for high-order Laplacians.
Understand? Yes. Yes. So, and is there anything known about what kind of properties of the brain you are looking at if you do something like a simplicial complex, or if you're not doing it, something more general than a simplicial complex, or is that too much asked?
No, it's a very important question. It's a question I'm asked in the hostel, because for many of my colleagues, it's like a black box, because there's a lot of mathematics. So if you just extrapolate, like in geometry, like, oh, in any dimension, it's just an extension, it's easy to explain. But of course, it's not only that, but in a way, the same way that we have hubs in node-wise, you can have hubs in three-point-wise. So all these notions that were built up in neuroscience over the last 20 years, if you say, oh, it's a geometric extension, like a high-dimensional extension of these concepts, you could come with some interpretation, but then I would have to enumerate in each direction, like for
yes.
But for the hubs is because it's related to what I just presented. Mathematically is nearly the same in every, like if you know how to extend in terms of dimension your metric, you can extend also the interpretation. Yeah.
Great, thank you.
Okay, I think we're on Claudia and JD.
I think my question is related to the question I was just asked. So you showed us the result for analysis with the three-body interactions, right? And you had those hub with the three, I mean, let's by three-body, I mean the three-body clicks. So you find the hubs. So if you only work with pairwise, you do not find this type of hubs.
Yes, no, we don't. Thanks for the question. So there's a different pattern. So I forgot to mention. Thanks, Claire, for the question. So it's basically when you do a normal hub analysis in the pairwise context, you get a list of nodes and the hubness, like the principal component, with which each coordinate has a value, and then you cannot distinguish without any, let's say, subjective criteria which nodes are hubs. You just say, oh, I want to take the 10 strongest ones as hubs, or the 5 strongest ones, or the top one. So there's no specific criteria to define a hub. And in that sense, if you choose the 10 strongest or the 20 strongest, you have different interpretation. And in this case, we just enumerate the hubs and they have a common property. They always share the same edge. That's why there's this, when you go to this, play this video, like when you go to this right side figure, there's a common feature. That's every triplet shares the same edge. And that is in this, you can distinguish this from the common pairwise framework. You would not find only one node has hub because you get just a list of hubness. In terms of statistics, it's the same pattern. Like when you do hubness analysis in the brain, you have lots of nodes with zero hubness and a bunch of nodes, let's say, I don't know, 10% of the nodes with high hubness, but you cannot tell, oh, this is the hub. And when you do in this approach, it's emergent. So you can, oh, actually the vision areas are higher the hubs. And I'm also not plotting one hub. I'm plotting now like 50 triangles. So there are 50 triplets that are important, but when you project, you actually say, oh, this is the hub from the vision. It's not like, I don't know if that answers your question.
If you were to use 4 body, for example, did you try that? So like removing, just consider only 4 bodies, because it's a different type of parity. So maybe it's maybe it would do nothing. I don't know.
That was nice. I did that 4 body. So you were a bit careful because it's very new. Then in both medicine and in the networks. So you want to report just in the first publication, the 3 body. But for 4 body, what happens, a more complex organization in the brain emerged. So if the brain lato sensu, there are many, there's no consensus, but the people split the brain in broadly in 7 big areas, like big systems. And the motor and the vision system are the simplest ones, like the primary systems that we have since evolution, like the primary animals also have that. But then you go to default modes, frontal parietal areas, or more frontal areas. These patterns emerge when you either increase the order, like when you ask the 4 order, we found the default mode network emerging, which is more complex than the vision and the motor area. And if you change to synergetic interactions, because I put the interaction information is redundant in this case that I showed, but if you change to synergetic interactions, we also change the hubness. But the logic is similar. What you find are triplets that share the same edge, and then you pop up, oh, these are actually is segregation in this area, like vision, and integration with the rest. So the triangles are making, or the polygons are making these. But yeah, thanks. And yeah, we are not in doubt whether to report this or not in the first work, but it's an important question.
Maybe one last about this. Do you understand? Is there some cases where you understand the choice of the metric and the connection between the choice of the metric that you have chosen and the hub that you find?
Yeah, that's also interesting because these two that I showed, like one is interaction information is with motor, and if you, it's a bit tough because it's more about having the data-driven analysis and then create a story, but it makes sense in a way, like because the interaction information, the intersection of the signals, let's say like that, and then because motor and sensory systems are really coupled, then this could be a way to capture that. And total correlations is mutual like feedback, like a total correlation which relates to vision. That could be a way, but I feel uncomfortable to reporting because it's like it's first work on that, so I cannot tell this is the ground truth. But yeah, you can try to draw interpretation. I would expect this more with time. Like if it's reproducible, I also test in different cohorts and different people ask to analyze in different schemes. So with time we may have this connection, but I feel if you follow the literature in brain networks, like in 2006 there was a paper, oh actually if you get Pearson correlation you define a network, and then it continued, and then no one knew the real interpretation. And over the last 15 years there were different metrics and interpretation for each metric. So if this becomes relevant, then this will come. I don't know if
yeah. Can I ask just one last thing just about the technique? So the picture that you're showing us right now, they are from average over different patients. Like, do you take data from different patients from the patient from the same cohort and then make the whole map, or do you make a map for each patient and then kind of create an average map? How does it?
We did both, so the variability is low, but some in an individual level, some people have different hubs, but the hub always exists this way. But what I report now is an average map, because like that when you go this high scale, like many cohorts, like big cohorts in neuroscience, you kind of create a mean of these connectivity metrics. So that's on the I'm reporting, but we also make a study in individual level, like. For each person, you can also make this analysis and check where are the hubs, and they fluctuate around what I report.
Okay.
Like some people, of course, people are different. So some people may have in rest state because there are multiple nodes that are related to vision. Let's say like that. If you go to this example, so it's not only. I think this one is the cuneus, but there are other areas, the occipital areas that are related to vision. So they have the high order hubs, but not necessarily the same. But there's a small variability, which is expected because it's like no one is equal. But in terms of making the message, we reporting in the mean.
Thanks.
Thanks.
There's another question on Zoom. I'm seeing JD. Please go ahead.
Hello. Thank you for the interesting presentation. So actually, I already got the answer to my questions, but I just I know calculating a higher order or multivariate information measures is time consuming, sometimes challenging. So I just wonder if we want to study the brain network more generally, not only triangle, but 4-body, 5-body. I just wonder if we can obtain the general network, a higher order network, from the pairwise, indirectly from the pairwise interaction network, because inferring pairwise interaction, I think, is easier than triangles, I think.
Yeah, it's a good question. Thanks for raising that. Yeah, so far I think up to 2 years ago in TDA, people would do what you say, like approximating high order interaction as a sum of PY interactions. This approximation is not that bad. It just, you may miss something. So then what we try to look for is what are we missing? So in a way, depending on what you're doing or your research question, it may be enough to go PYs, but in terms of what we are now finding, that these are kind of difference between the PYs and the high order. In terms of the complexity, lots of is going on in terms of optimizing this process. If you follow the work of Rosas and Rodrigo Coffey and other authors, they did a few papers already in the brain on high order interactions. They just don't put the network structure in the interaction. So they only measure the high order connectivity without creating a high order network. And for their papers, if you send an email, I can send those papers. They go up to 20, order 20, but then they created the atlas that is really coarse. So they analyze the atlas with much less nodes, because basically if you have n nodes, you have 2 to n possible interactions. So it's really exponentially hard. And as a physicist, I would say, okay, each order will contribute, but each order will contribute a bit less in a way. So I think 3-point interaction is meaningful, 4 or 5, but I would not expect like a 20 order interaction to be relevant. So I think it's still in an exploratory phase. And I agree with you as a limitation combinatorially, at least until you have a quantum computer. But I think it works still to do up to a few orders or decrease the resolution of your atlas. So if you decrease the resolution of your atlas, you can do a lot. But the brain atlas from 20 nodes up to 1,000 nodes. So this choice is also tough. Like in neuroscience, there are many choices. But yeah, to address this, either less orders or less smaller, like more coarse atlas.
Okay, thank you.
You're welcome. My pleasure.
All right, great. Do you have any questions here in the room still?
Thank you. I'm happy.
A lot of questions. Well, then I guess we can thank Fernando again.